So I'm going to talk about genome editing to enhance reproductive capacity in livestock, some of the work we've been doing in this space for more than a decade now. And so our guiding principle for improving reproductive capacity is that genetic gain to achieve trait improvements for feeding people relies on the propagation of trait-driving genetics through germ cells, which are the conduit of genetic information flow across generations. And so devising of novel reproductive biotechnologies that will enable widespread access and large-scale dissemination of elite or gene-edited genetics can have a massive impact on production efficiency in livestock. So our approach to trying to develop this principle focuses on biotechnologies that capitalize on the male germline and on the power of spermatogenesis to produce millions of sperm daily, all of which carry potentially trait-driving genetics. In all animals, the foundation for spermatogenesis is provided by the actions of a stem cell population, often referred to as spermatogonial stem cells or spermatogenic stem cells. These cells are a subset of the spermatogonial population, and this population is derived postnatally from precursor cells that are referred to as prospermatogonia. And these prospermatogonia arose in fetal development from primordial germ cells that have been specified in an early embryo. And so the capacity to establish the sperm producing lineage is laid down during this developmental trajectory. And it is in the postnatal spermatogonial stem cell population that has the greatest potency for driving large scale spermatogenesis. And so methods for isolating and culturing and genetically modifying and then transplanting spermatogonial stem cells have held massive potential for enhancing reproductive capacity in livestock. And the first indication that these cells might have this power to be able to influence trait-driving genetic gain in livestock production was really pioneering studies in mice by Ralph Brinster's group in the 1990s. And they demonstrated the functional capacity of spermatogonial stem cells to be isolated from testicular tissue of a donor male and either transplanted as a fresh cell population or following a period of in vitro maintenance into the testicles of a germ cell depleted recipient male. And in these studies, the depletion method was to use a chemical chemotoxic agent to deplete the germline, which didn't ablate the germline. And so there was still endogenous spermatogenesis that could occur in the recipient testis. But some of the donor stem cells were able to engraft and produce colonies of sperm production. And these mice could actually mate by natural breeding and sire offspring that had the donor genetics. And so for the better part of 2 decades now, my group and other groups around the world have been trying to translate this idea of spermatogonial stem cell isolation, culture, and transplantation from mice to livestock as a reproductive biotechnology to enhance the dissemination of elite genetics. Our work in this area really started roughly 2004, and this was well before gene editing was on the scene. And we struggled to find ways to ablate the germline of a recipient male while keeping the testicles functionally intact. And so when CRISPR-Cas9 was adapted for mammals, we started working towards an application that we now term surrogate sires. We believe this technology has the capacity to be a breeding tool for efficient propagation and dissemination of either elite or gene-edited genetics across the spectrum of livestock production. And we've been working to develop this for cattle and pigs, as well as small ruminants. So I'm going to use beef cattle to portray the overarching concept, but you could envision how this could apply really to any livestock production system. So the idea is that spermatogenic stem cells can be isolated from testicular tissue of an elite, or it could be a gene-edited donor male and used freshly, or they could be cryopreserved, or they could be put into a culture dish for in vitro expansion of numbers, and then transplanted into the testes of surrogate recipient bulls. And the donor stem cells would engraft and generate sperm that possess the donor genetics. And so with a battery of the recipients generated, they can be placed in a conventional breeding scheme to sire progeny with donor genetics through natural mating. And so a couple key aspects to developing this type of concept are in vitro, the ability to in vitro expand donor stem cells, and we've worked in this area, and I'll talk a little bit about it here in a couple minutes. And also the ability to create germline ablated recipient males that can't make their own sperm, but still have functionally intact testes. And so for many years, we were struggling on how to answer this question of how do we generate male livestock that don't make their own sperm, but still have functional testicles. And so our approach has been to use CRISPR-Cas9 gene editing to target key genes that are needed just for male germ cell survival. And by doing that, we can knock out the male germline. And this is important to target male germ cell survival, because if we were engineering alleles that resulted in germline ablation, then in a homozygosity state, we would have sterile males that could be surrogate sires, but the female germline would be intact. So females would still be fertile even in a homozygous state. And then in a heterozygous state, the males would still be fertile because of one functional copy of the key gene, and females would be fertile. And so you could envision a generator herd could be produced where we can simply cross homozygous knockout females with heterozygous knockout males to generate more homozygous sterile males that could be used as surrogate sires without having to go back and edit embryos over and over. And so our target gene has been the nanos2 gene. And this is because work in lower order organisms like flies many years ago had identified a single nanos gene and demonstrated that knockout of that gene led to both male and female sterility. But the flies were otherwise normal. Mammals have 3 nanos genes, and work with ES cell gene targeting in mice had demonstrated that knockout of the nanos2 gene resulted in male sterility, but females were fertile. And so we started in this area in roughly 2013 in CRISPR editing of the nanos2 gene. And at that time, we didn't know the effect of nanos2 knockout in other species other than a mouse. And we also didn't know whether a nanos2 knockout male could become fertile if we reintroduced germline. And so to address these questions, but also to establish a proof of concept pipeline for a surrogate sires breeding technology, we set up this type of workflow where we would use CRISPR-Cas9 to generate nanos2 knockout alleles. We'd produce knockout males and phenotype them for whether they had germline ablation or not. If they did have germline ablation, we would then transplant donor sperm stem cells into their testes and try to answer the question of whether their testes supported donor-derived sperm production. And if they did, could we refine the whole process to where we could now get natural fertility? And so we've been working to apply this across a spectrum of animals, including a model organism in the mouse. I always say if you can't achieve it in the mouse, it's unlikely you can achieve it in other species. And so we developed proof of concept initially with the mouse and then extended this to livestock, including pigs, goats, and cattle. A lot of the work has been published in an article in 2020, but some of the work with cattle is currently unpublished, but we're working to get this published hopefully within the next year. And so with mice, we've been able to achieve all 5 boxes. We can refine the whole process to where mice are naturally fertile and sire offspring that have donor genetics. In pigs and goats, we've been able to, at this point, check 4 of the boxes, and we're now working on refinements to get them naturally fertile. In cattle, we've been able to check all 5 boxes as well, where we can generate surrogate bulls that are naturally fertile. And again, some of this is unpublished work that hopefully will be out soon. And so I want to transition here for the next couple of minutes to talk about what I see as the next frontier as we start to refine the surrogate sires technology to be a applicable tool in livestock production. And what I see as the next frontier, what's up and emerging, is incorporating surrogate sires with a strategy to do direct germline gene editing. And so I'll come back to cattle again to demonstrate portray the overarching concept. So the idea is that if we can take stem cells from testicular tissue of an elite male or desirably genetic male, maintain them in a culture dish for an extended period of time, we can introduce CRISPR-Cas9 components and edit that male's germline directly. And then go through a selection process where we're choosing stem cells that have the intended edits, clonally expand those stem cells where we can do deep screening of both intended and unintended edits even before we generate an animal, and then collect those stem cells, transfer them into the testes of Nanos 2 knockout surrogate recipients, and allow them to breed by natural mating in a conventional breeding program and generate progeny that have the donor elite genetics plus whatever gene edits have been introduced in the cultured stem cells. So some of the key aspects to achieve in this type of concept is we need to be able to have conditions that support exponential growth of SSCs. We need to be able to efficiently deliver gene editing components into these stem cells in a culture dish, and we need to be able to have a strategy for clonal expansion of the cells. And so I underline actual as part of the SSCs because lots of cells from testicular tissue can grow in a culture dish, and sometimes they look similar, but the definition of an SSC is the ability to generate sperm. And so just being able to observe something growing in a culture dish doesn't tell you anything about its stem cell capacity. So we've been working on a proof of concept pipeline across the mouse, pig, cattle, and sheep to develop stem cell culture conditions, spermatogenic stem cell culture conditions. And some of the work has been published, and some is still unpublished at this point. But for all of these species, we've been able to develop strategies for efficient isolation of stem cells out of testicular tissue. We've optimized the in vitro conditions to where we can get long-term, multi-month maintenance of the cells in a culture dish. In the case of mice and pigs, we're able to get exponential expansion. These cells grow quite rapidly for a germ cell in the culture dish. In cattle and sheep, they can grow, but they don't grow in an exponential growth phase. So we're still working to refine those conditions. In all the species, we can show they express telltale biomarkers of germ cells, as well as an undifferentiated spermatogonia. I put an asterisk by biomarker expression because just expressing a biomarker doesn't tell you much about the functional capacity of a cell. And when it comes to a spermatogenic stem cell, the unequivocal evidence is if those cells can make sperm. And to do that, they have to go back into testicular tissue to demonstrate they can generate sperm. So we've been able to demonstrate this for mice and pigs, that what we grow in the culture dish have bona fide stem cell capacity for making sperm. We're still working to demonstrate that in cattle and sheep, but hopefully soon. And so we've taken this one step further in the mouse as a proof of concept to answer the question of whether we can derive offspring with an engineered trait by CRISPR-Cas editing of cultured spermatogenic stem cells. And so in this workflow, we use a wild type donor mouse, isolate stem cells from testicular tissue, put them in a culture dish, and introduce CRISPR-Cas9 components for driving an edited trait. Transfer those, and in this case, we didn't have clonal expansion at the time, so we were transplanting a mixture of things that have been edited and unedited, but put them into a Nanos 2 knockout recipient. It can't make its own sperm, but can only make sperm from what we transplant in. Breed that with wild type female mouse and ask, can we get a CRISPR modified offspring on the back end? So the trait we went after was something that would produce a short-tailed phenotype in the mice because it's easily observable. and we are hypothesizing that it could be heterozygous dominant, so it wouldn't have to have a homozygous editing or biallelic editing condition in order to observe a phenotype. And so we've run this through several times now, and this is just an outcome of one of those where we can get germline, direct germline edited offspring with short tails by editing the spermatogenic stem cell in a culture dish and combining it with surrogate sires technology. So we're getting close on surrogate sires, and I think this direct germline gene editing is something that can be really powerful. And so I want to go through just a couple of implications here. We think that this type of technology will be able to empower efficient generation of gene-edited livestock germplasm, and that it enables characterization of both intended and unintended DNA editing prior to ever even generating the animal. Because if we can clonally expand the edited stem cells, we can do deep characterization in a culture dish before we even try to create a gamete and generate an animal off of it. It also, we believe, will provide a simpler dissemination of edited genetics into production populations, because now we could generate a battery of recipient males. They're all carrying already edited germline, and this will enable better deployment. So we do have challenges and barriers in trying to overcome to bring this to a utilizable tool in livestock production. One of those is the ability to scale and customize for Nano's 2 knockout sires. The scalability of generating animals is a challenge, but with a generator herd with knockout females and heterozygous males, it should be enabling. The challenge is developing it for multiple different breeds based on what a producer might want. We also need to we're also working on optimization and refinement of spermatogenic stem cell culture conditions, as well as spermatogonial stem cell transplantation. Depending on species, there's little tweaks that need to be made. They can be technically challenging and require a lot of research and development. And then lastly, there's an issue of affordability and harmonization of global regulatory frameworks for us to truly be able to integrate this into production systems globally. The cost that's needed for seeking approval as well as commercialization is a little bit prohibitive at the moment, and the lack of having harmonization of what the regulatory landscape looks like between countries is also a challenge. So I think I'll close there and just say thanks for tuning in, and I'm happy to answer questions in the Q&A session. I'm also happy to answer questions that you may want to send me by email. This is my email address. Please feel free to contact me. Thanks.